The world in which we live can be divided into two very basic parts. Things that are alive, such as these microorganisms, and things that are not, such as this rock. Among living things, there are unbelievable variations. But as strange as it seems, this sea anemone, this elephant, these tiny rod-shaped bacteria, this crab, and both this butterfly and flowering plant all share certain traits that clearly distinguish them from non-living things. Scientists call these shared traits the characteristics of life. The first characteristic of life is that living things can reproduce. This means that, like these giraffes, they can produce new life that is very similar to themselves. The second characteristic of life is that, like these young fish, all living things can grow and develop, forming mature organisms that are capable of reproduction. The third characteristic of living things is that they can respond to the outside world. For example, these striped fish are responding to the much larger whitefish by getting out of its way. And this bee has sensed the pollen in this flower and is responding by gathering it to make honey. Being able to respond to things in the outside world is very useful because it helps living things find food, protect themselves from harm, find mates, and perform many other important activities. The fourth characteristic of life is that living things need to use energy to stay alive. For example, plants such as these grasses can trap the energy of sunlight, and this energy not only keeps the grasses alive, some of it is chemically stored in their leaves and stems, so that when animals like these cattle graze, they are able to get the energy they need to power their life processes. The fifth characteristic of life is that living things can change as their environments change. And that is because life has been able to change and adapt over millions of years to meet some very extreme conditions, that living things can now be found in almost every imaginable place on Earth, such as the algae that live in the boiling water of this hot springs pool, or the dry lichens that cover the surfaces of these rocks, or the bacteria that thrive in the human mouth and digestive tract. The sixth characteristic of life is that living things can move. This characteristic is obvious when movement is fairly rapid, but it is harder to see when it is slow, such as the movements made by plants as they turn their leaves toward the sun. When it comes to life, some creatures are very complicated, while others are quite simple. But no matter how simple or how complicated they may be, living things are always very highly organized in the way they are put together. In fact, it is because they are so highly organized that scientists usually refer to living things as organisms. If we look closely at how living things are organized, one fact stands out. They are all made up of cells. For example, the tips of these tiny onion roots, when stained and viewed under a microscope, look like this, a collection of tiny compartments called cells. Cells are defined as the simplest structures that can carry out all of the activities characteristic of life. That is, can reproduce, grow and develop, use energy, and so on. And because life cannot exist without them, scientists call cells the basic units of life. The tiny microscopic organisms seen here have only one cell. They are called single-celled or unicellular organisms, and are examples of some of the simplest living things. But as we saw in the case of the onion, as living things get bigger, they will always have more cells. In fact, some organisms have hundreds of trillions of cells of many different types. Creatures made up of more than one cell are called multicellular organisms. Within the bodies of multicellular organisms, cells have become specialized to carry out specific tasks. For example, these red blood cells are specialized to carry oxygen to other cells all over the body, while this white blood cell, stained with purple dye, is specialized to kill the harmful bacteria that cause certain diseases. Multicellular organisms can have hundreds of different types of specialized cells, and yet it is quite rare to find cells that carry out their tasks alone. That is because in complex organisms, cells are almost always found working together in organized groups. For example, layers of cells that are similar in structure and function are called tissues. Here we see a tissue of cells that makes up the outer layer of a leaf. Very often, several different types of tissues are found grouped together to form organs, as we can see here in a slice of a disease-fighting organ called a lymph node. Beyond that, certain organs combine to form organ systems, which when taken together create a whole organism. The human organism has 9 basic organ systems. The first is the skeletal system, which gives the body support and helps protect the delicate internal organs. Bones are the main organs of the skeletal system. The second is the muscular system, which allows the body to move. The muscles are the main organs of this organ system. The third is the nervous system, which senses the environment, receives information from the body, helps coordinate physical activities, and makes it possible to think. The brain is the main organ of the nervous system. Fourth is the circulatory system, for carrying blood to cells throughout the body. The heart is the main organ of the circulatory system. Fifth is the excretory system, which allows the body to rid itself of liquid waste. The kidneys are the main organ of the excretory system. Sixth is the digestive system, which allows the body to process and absorb food and water. The stomach, small intestine, liver, pancreas, and large intestine are all important digestive organs. Seventh is the endocrine system, which is made up of different glands that produce the chemical messengers called hormones. Among the organs in the endocrine system are the thyroid, adrenal, and pituitary glands. Eighth is the respiratory system, which provides the body with oxygen and helps it get rid of the waste product, carbon dioxide. The lungs are the main respiratory organs. The ninth and last organ system is the reproductive system, which allows us to have children. The female ovaries and the male testes are the main organs of the reproductive system. As we have seen, all 9 of these complicated organ systems are made up of organs that are made up of tissue layers of specialized cells. In the next section of this program, we will look inside a specialized cell and learn how it works. The basic units of life are called two characteristics of living things are that they can and creatures seen here are called because they have only one cell. Bigger, more complicated creatures will have many more than simpler ones. Because they are highly organized in the way they are put together, living things are often called similar kinds of cells usually form layers called in multicellular organisms, cells have become to perform certain tasks. The organ system used to pump blood is called the system. The organ system for breathing is called the system. The brain is the main organ of the system. All cells, including the single-celled amoeba seen here, are made up of a living jelly-like substance called protoplasm. The protoplasm of a cell is always wrapped in a flexible skin called the cell membrane. The cell membrane helps control what comes into and what goes out of the cell. What comes into the cell are such things as oxygen, water, and food. What goes out are wastes and special cell products. As can be seen in this stained non-living nerve cell, the protoplasm inside the cell membrane is made up of two basic parts, a rounded nucleus and the cytoplasm, which is all the protoplasm outside the nucleus. As you can see by looking at this living cytoplasm, it is very complex. It is always in motion and contains lots of tiny organs, or organelles, plus other structures that carry out a variety of tasks inside the cell. Now let us look at some simplified diagrams of cells to learn about the components of the cytoplasm and how they function, such as the mitochondria, the powerhouses of the cell, where stored energy is released. The lysosomes, that contain digestive enzymes. The food vacuoles, where large food particles are stored and digested. The centrioles, that are important in cell reproduction. The ribosomes, where proteins are made. And the network of hollow tubes upon which most of the ribosomes sit, called the endoplasmic reticulum, or ER, used in transporting proteins inside the cell. The Golgi complex, where proteins are packaged. And in plant cells, the chloroplasts, which are extra important organelles because they are used to create food. Here we see thousands of green-colored chloroplasts in the cytoplasm of a living cell. These chloroplasts are busy carrying out a process called photosynthesis that is essential to nearly all life on Earth. Inside the chloroplasts, light energy is used to combine carbon dioxide gas, CO2, and water, H2O, to make sugar. As a result of photosynthesis, the sun's energy actually becomes a part of the sugar's chemical structure. This is why humans can obtain quick energy by eating foods containing sugar. Now let us look at how the different parts of a human cell work together to use the sugar and other food materials. After being eaten, food travels down the digestive tract, through the stomach, and into the small intestine, the part of the body where food is first taken into cells. Here, the sugar in the food is simply carried across the cell membranes of the cells that line the small intestine. This is possible because sugar is not a very large chemical substance. However, other materials in the food may be too large to enter the cell in such a simple fashion. They will be surrounded by the cell membrane, which pinches off to form tiny sacs called food vacuoles, a process called phagocytosis, or cell eating. The food vacuoles will then be entered by lysosomes, which are little more than packages of digestive enzymes. As a result, the food inside the vacuoles will be broken down into useful nutrients. The lysosomes, you will notice, are created by another organelle called the Golgi complex, which is the place where the digestive enzymes are wrapped in a special membrane. Some cells contain thousands of these organelles. By the time the food in the food vacuoles has been broken down into nutrients and released into the cytoplasm, the sugar will have already reached the organelles called mitochondria, the powerhouses of the cell, which can sometimes number up to 2,000 per cell. Once the sugar gets inside the mitochondria, it will be chemically combined with oxygen to release the sun-stored energy. This process, called cellular respiration, also creates two waste products, water and carbon dioxide. It is because our cells need oxygen to release chemically stored energy that humans breathe in oxygen and breathe out carbon dioxide. And the more energy we use up, the faster we must breathe to take in the extra oxygen needed to release more energy. In these pictures of a living white blood cell, the mitochondria can be seen moving about in the energetic, life-supporting dance that accompanies cellular respiration. It may not be obvious, but cellular respiration is a lot like a burning fire. For when a fire burns, oxygen is used up, carbon dioxide and water are produced, and energy is released. The same thing that happens during cellular respiration. But it is important to realize that sugar is not actually burned inside the cell. Instead, the energy stored in sugar is released through a series of complicated steps that produce a special high-energy chemical called ATP, or adenosine triphosphate. Cells use the readily available energy of ATP in much the same way that cars use gasoline. And without a doubt, one of the most important of the cell's energy-consuming activities is making proteins. Proteins are made on the millions of tiny organelles called ribosomes, organelles that themselves are produced in a special part of the nucleus called the nucleolus. And because most ribosomes are found attached to a complicated network of passageways called the endoplasmic reticulum, or ER, newly made proteins can be easily transported through these passageways from one part of the cell to another. Here we see a picture of the endoplasmic reticulum and ribosomes taken by a powerful electron microscope. The reason that proteins are so important is that they carry out many different roles. Some are used to build and repair cells. Some help fight disease. Others help transport particles across the cell membrane. And nearly every time chemicals need to be combined or changed inside cells, the proteins known as enzymes must be present, or the chemical reactions will not take place. Some types of cells, such as those that line the small intestine, even make enzymes that are used outside the cell. These proteins go through the ER to the Golgi complex, where they are specially prepared before being shipped into the digestive tube. But most of the proteins made on the ribosomes are used inside the cell. Some proteins are needed in the nucleus. And to get to the nucleus, these proteins move through the ER until they pass through the nuclear membrane. The nucleus is the control center of the cell, and inside the nucleus, some of the proteins made in the cytoplasm help create structures that guide the activities of the cell. For example, the living nucleus we see here is composed mostly of a protein-containing substance called chromatin. Normally, the chromatin doesn't look like much. In fact, it only begins to really look like something when the cell gets ready to reproduce. Then the chromatin condenses into the worm-like structures called chromosomes seen here. And even though part of each chromosome is protein, a lot of it is a very complicated chemical called DNA, or deoxyribonucleic acid. Human cells have 23 pairs of chromosomes. Here we can see exactly how the DNA is arranged inside a chromosome, for each tiny human cell contains enough tightly folded DNA inside its chromosomes that if all of it were stretched out, it would be over 6 feet long. DNA is the most important chemical in the entire cell, because the DNA is where the information on how to run the cell is stored. The DNA in the chromosomes is made up of 2 long, twisted strands, each of which is built from 4 small subunits containing the chemicals adenine, guanine, cytosine, and thymine, which scientists have abbreviated AGTC. The cell puts these units together in a definite order to create an amazing chemical language called the genetic code that is used by all living things to store information. In certain ways, the genetic code resembles a written language, but the only letters in its very short alphabet are the chemicals A, G, T, C. And even odder is the fact that the 4 letters in its alphabet can only be used to make words that are 3 letters long, so that the entire genetic code dictionary contains just 64 different words. Scientists have discovered that, except for 3 or 4 punctuation words, all the rest of the genetic code words in DNA represent the chemical building blocks called amino acids from which proteins are made. This means that the long chains of DNA in the chromosomes are nothing more than plans that tell the cell how to make different proteins. And no matter whether the proteins are put to use in a human cell like this one, or in simple one-celled organisms like these, the proteins will do what is needed to keep the cell alive. This means building and repairing old or worn-out parts, transporting particles, helping fight disease, or by acting as enzymes that allow chemicals to combine inside the cell. Each set of genetic code plans for a protein is called a gene, and genes, through the proteins they make, help determine physical characteristics, such things as the color of skin, eyes, and hair. Genes are arranged on the DNA one after another inside the chromosomes. And today, scientists working on a huge scientific research project called the Human Genome Project, are busy mapping the exact location of each of the approximately 100,000 different human genes. In the next section of this program, we will find out what happens to DNA, genes, and chromosomes when cells reproduce. Sugar is one of the main sources for cells. The organelles called are also known as the powerhouses of the cell. Tens of thousands of tiny protein factories are found on the surface of much of the cell's endoplasmic reticulum. The organelles are made in the nucleus in a special area called the nucleolus. Much of the material in the nucleus is used for storing the language used by cells is known as the chromosomes contain a long, twisted, thread-like chemical called chromosomes are not always visible in cells. They can be seen only when cells are getting ready to The cell's language uses a chemical alphabet that is only letters long. Each set of protein plans is called a As we learned in the first part of this program, the ability to reproduce is one of the most basic characteristics of living things. Microscopic one-celled organisms, such as those seen here, usually reproduce by asexual reproduction, by simply dividing and making two new cells. With asexual reproduction, only one parent is needed to create offspring. But large multicellular organisms usually have two parents. They develop from the fertilized egg created when male and female sex cells join during sexual reproduction. Even though large organisms often require two parents to produce offspring, their individual cells reproduce using a method that is like asexual reproduction in one-celled creatures. Let's find out how this works in an animal cell. Before a cell can reproduce, it begins by reproducing its nucleus, a process scientists call mitosis. The first and longest stage of mitosis is called interphase, a period of cell growth when the nucleus is filled with chromatin fibers. During interphase, a process called DNA replication takes place. The old DNA is copied, and two new sets of DNA plans are made. The replication of the DNA results in a duplication of the chromosomes. But before the duplicated chromosomes appear from the chromatin, the centriole pairs are also duplicated. and early in the next stage of mitosis, called prophase, the centrioles move to opposite sides of the nucleus, where tubular structures, called spindle fibers, form around them as the newly duplicated chromosomes come into view. During the next stage of mitosis, called metaphase, the duplicated chromosomes line up in the middle of the spindle fibers. Then, during the fourth stage of mitosis, called anaphase, the spindle fibers shorten, and each duplicated chromosome is pulled apart, creating two separate chromosomes. During telophase, the last stage of mitosis, two new nuclei form where the chromosomes have gathered, and soon two new daughter cells are created after the cytoplasm divides by a process called cytokinesis. The new daughter cells are now back in interphase, and each will grow until it reaches the size of the original parent cell. Now let us repeat our look at mitosis and cell division without stopping, beginning with interphase, to prophase, to metaphase, to anaphase, and finally to telophase and cytokinesis. And now, let's follow the cell reproduction process in a living cell. First is interphase, then prophase, then metaphase, anaphase, telophase, and cytokinesis. In multicellular organisms, new cells are normally only produced when old or damaged cells need to be replaced, or when a young organism is growing. But it is interesting to note that cells occasionally lose the ability to control how fast they reproduce. These cells, called cancer cells, just keep reproducing over and over again until the organism is finally destroyed. In human cells, out of a total of 46 chromosomes, one set of 23 chromosomes comes from the male parent, and the other set of 23 comes from the female parent. This means that human cells normally have two complete sets of chromosomes. Having two sets of chromosomes is called the diploid condition of the cell. But in certain cells, called germ cells found only in the testes of males or the ovaries of females, a process called meiosis takes place that produces haploid cells, cells that have only half the normal diploid number of chromosomes. These cells are called sex cells, the sperm and eggs. Now let us follow the process of meiosis step by step. Like mitosis, meiosis begins with cells that are in interphase. No chromosomes are visible, but the DNA is replicated during this stage. The next stage of meiosis, called the first prophase, is quite different from mitosis because the identical pairs of duplicated chromosomes start to group together. And during the first metaphase stage, the duplicated chromosome pairs line up in the center of the cell facing each other. During the first anaphase of meiosis, the duplicated chromosomes don't split apart like they did in mitosis. Instead, they stay stuck together, and one set of each duplicated chromosome pair is pulled in an opposite direction, so that after the first telophase, single duplicated chromosomes remain in each daughter cell. If this were mitosis, we would be finished at this point. But in meiosis, we have only reached the halfway mark. Now the new cells are in a very brief interphase in which the chromosomes only partially retreat back into chromatin. And very importantly, the DNA is not replicated before the daughter cells enter a second prophase. From now on out, everything occurs just as it would during normal mitosis. Having reached the end of meiosis, we can see that, compared to mitosis, a big change has taken place, because each of the new sex cells has only half the number of chromosomes of the original germ cells. Now let us look at meiosis from beginning to end without stopping. As the germ cell enters the first prophase, the first metaphase, the first anaphase, the first telophase, and the first cytokinesis. Then the new cells enter the second prophase, the second metaphase, the second anaphase, the second telophase, the second cytokinesis. Even though this example demonstrates the typical process of meiosis in multicelled organisms, it should be pointed out that owing to unequal cell divisions in human females, only one functional egg is produced by meiosis. Nevertheless, if the sex cells produced in this example were human, they would each contain a haploid number of 23 chromosomes. So that, if the egg and sperm were to join during fertilization, they would create a fertilized egg having 46 chromosomes. And from this fertilized egg, through repeated cell divisions, a diploid human embryo would develop. One further fact to point out is that mistakes sometimes occur when germ cell DNA is replicated. Mistakes such as the one seen here, in which a genetic code letter is accidentally deleted. Such mistakes are called genetic mutations. They create inaccurate cell information. Having the wrong cell information can cause terrible genetic diseases, such as Huntington's disease, which has afflicted many members of this family. This serious genetic disease is not often seen, but the genetic diseases cystic fibrosis and sickle cell anemia are quite common and afflict millions of people worldwide. Yet it is important to note that genetic mutations are not always bad. For in extremely rare instances, they actually have positive effects that give an organism a better chance of surviving. And it is because of these good genetic mutations that organisms have been able to adapt and change over the centuries as their environments have changed. Before cells divide, the nucleus has to reproduce. This process is called DNA replication. Takes place during the first stage of nuclear reproduction, called sex cells. Have only the number of chromosomes of body cells. Fibers called form around the centrioles of animal cells during the reproduction of the nucleus. The process in which male and female sex cells join together is called